Forskningen kring varför vissa människor blir schizofrena eller manodepressiva går framåt tack vare genteknik. Och nya effektivare mediciner kan tas fram. En av världens toppforskare inom området är Hyman från Broad Institute på MIT.
The problem in making models is that the traditional model organism, which is the mouse, while very useful for basic science, diverges too much in evolution from humans, especially in recent, evolutionarily recent brain areas. And the other thing about making transgenic mice is it's not high throughput. And what we're discovering is that there are many hundreds of genetic variants we have to investigate. And so we've begun to use human stem cells in vitro to engineer them and to turn them into neurons.
So and you implant them in the mice then, or?
We have not. It's an idea to implant them in the mice, but right now what we're really trying to do is to make really good human, say, pyramidal neurons or interneurons, kinds of neurons that have been implicated in schizophrenia, in autism, and grow them and study them in a dish. Ultimately, there are other things you can do. You can grow them, let them grow into little organoids, little tiny brainoids. They don't get too big because they don't have a blood supply. And some people have advocated actually implanting them in mice and letting them grow.
It's interesting. But what's the problem with today's drugs?
Well, the drugs that we have are very useful, but we've simply run into a brick wall. The drugs haven't significantly improved in about 50 years, and we've run out of clues. Now, the genetics is giving us important brand new clues, but a list of genes doesn't tell you how to develop new drugs. In the end, we need models that we can use for both understanding and for screening, and we're hoping that these engineered stem cell derived neurons will be useful models for making new medicines.
Are there any other tools you can see that point forward, so to speak?
Well, there are certainly other important tools that we're going to use. Some human neurotechnology might be useful in the sense that if you create an abnormality in a cell by putting in a disease gene and you find out what that does, creates a phenotype, that is, the cell behaves in a certain way, the question is, does that matter at all to a human patient? And so for the most promising findings, we're going to need to go from a particular finding or phenotype in a cell, perhaps to an animal model, but to something that translates to a human. So technologies to measure brain rhythms, brain imaging that can go back and forth will be absolutely critical.
You said in your speech that pharma industry is exiting psychiatry.
Yes.
And what do you think about that? Is there research now being done, the important research being done at institutes and universities like yours?
Yeah, I think so. I mean, in the end, we need pharmaceutical companies to succeed because they have the resources to make medicines, and especially the later stage issues, looking for toxicity, doing large clinical trials, academia really cannot do. It's too expensive, and we're less skillful at that, except partnering perhaps with a big company. But because they are exiting, saying it's just too hard for now, a lot of the more fundamental research is occurring in universities and hospitals.
But maybe one needs new business models.
Perhaps we need new business models. I think that if we start to succeed, there will be commercial opportunities and new business models might very well form. But I think in terms of turning this ultimately into a business, we have to be a little bit farther along in turning this important emerging genetic information into useful screening tools for drugs.